Good day. The topic to be discussed will be the histology of the cell. The lecture is divided into two videos. The first video will be the lecture on the general characteristics of the cell and the nucleus. The second video will deal with the organelles and inclusion bodies found in the cytoplasm. Histology is the study of cells and extracellular components and their organization into tissues, organs, and organ systems. It employs more commonly the use of the light microscope. This shows two neutrophils and erythrocytes under the light microscope. Another instrument to study histology is the electron microscope, which are of two types. In the scanning electron microscope, the beam of electrons passes over the surface of the specimen. This is a scanning electron micrograph of a neutrophil. In the transmission electron microscope, the beam passes through the specimen, showing more details of the cellular structure. This is a transmission electron micrograph of a neutrophil. The most common instrument used to study histology is the light microscope, which uses light to magnify the image. One important parameter in microscopy is magnification, or the process of enlarging an object than what it is in real life. There are two parts in the microscope responsible for magnifying the image being viewed. The eyepiece, which is the first point of magnification, has 10 times magnification. The second point will be the objective lens. There are usually four objective lenses: scanning lens that magnifies 4 times, low power objective lens 10 times, high power objective lens 40 times, and oil immersion lens 97 to 100 times. To get the total magnification, multiply the eyepiece magnification by the magnification of the objective lens used. For example, when you use a low power objective lens, this will give a total magnification of 100 times. Even if the student is prevented from using the microscope now, it is important to know the parts of the microscope and its function. For more information on the light microscope, I have included a link of a video in your course materials. Microscopic units are used to describe the sizes of objects seen under the microscope. The first two units, micron and millimicron, are used for the light microscope. Angstrom is the unit of measurement used for the electron microscope. The cell is the basic structural and functional unit of all organisms. Similar cells are grouped into tissues, examples of which are muscle, bone, epithelium, and connective tissue. Groups of tissues make up organs. An example will be the trachea. The trachea has several basic tissues such as the epithelium, connective tissue, muscle, and cartilage. Organs with similar functions are grouped into organ systems. The respiratory system is composed of the nose, pharynx, larynx, trachea, bronchi, and lungs. Different cells within a single organism comes in different shapes and sizes depending on their specific functions and content. The cuboidal cells in the renal tubules are specialized to perform selective secretion and reabsorption to form urine. The flask-shaped Purkinje cell in the cerebellum has apical dendrites which receive impulses from other neurons. Smooth muscle cells are spindle-shaped, facilitating contraction. The oocyte is large and rounded, containing a lot of cytoplasm. The columnar cells lining the digestive tract has a secretory function. Cells are enclosed in a cell membrane and are partitioned into two major compartments, the nucleus and the cytoplasm, surrounding the nucleus and making the greater part of the cell volume. The nucleus contains deoxyribose nucleic acid that is responsible for encoding the instructions needed for the synthesis of all cellular proteins and enzymes that induce metabolic reactions in the cell. This is visible in the light microscope. It is in the cytoplasm that these metabolic processes take place. There are two main structures in the cytoplasm: organelles and inclusion bodies, which are both suspended in a gel-like matrix called the cytosol. The organelles are membrane-bounded and are involved in essential metabolic processes such as energy generation, protein synthesis, and other vital functions. Inclusions are stores of lipid and inert deposits of waste products from cellular metabolism. The cell membrane separates the cytoplasm from the external environment, forming the outer boundary of the cell. It is not resolved in the light microscope, but in the electron microscope it appears as a pair of parallel dense lines separated by a less dense intermediate layer. The phospholipids forms the basic structure of the plasma membrane. It consists of hydrophilic head portion, which are soluble with water, and a hydrophobic tail portion, soluble only in fats and cannot mix with water. The phospholipid forms a bilayer. In each layer, the lipid molecules are arranged in such a way that the hydrophilic head portion are at the outer and inner surface of the cell membrane, and the hydrophobic tails are located inside. The lipid constituent of the membrane is responsible for the cellular shape and its permeability properties. Precisely which substances can enter or leave the cytoplasm are determined by the property of permeability. Water, oxygen, nitrogen, and small uncharged polar molecules can cross the cell membrane without difficulty, but larger molecules such as glucose and amino acids are unable to diffuse through. Plasma membranes contain protein, which are of two types. Integral or intrinsic proteins extend through the bilayer and their ends are exposed on its outer and inner surface. Proteins that are bound to the outer and inner surfaces are called the peripheral or extrinsic proteins. These proteins act as carrier proteins, channel proteins, or receptor proteins. The glycocalyx are carbohydrate chains attached to proteins or the phospholipid molecules. It forms a coat on the outer surface of the cell. It has many functions. It can recognize cells based on the structure and type of carbohydrate in the glycocalyx and determine if they should be there or not. It binds cells together. It provides cushioning and protection for the plasma membrane. It enables the immune system to recognize and selectively attack foreign organisms. The nucleus contains the blueprint of all cell structures and activities. It is the command center of the cell since it contains the repository of instructions for protein synthesis and site of RNA synthesis. It is spherical or ovoid in shape. In the stained histological sections examined under the light microscope, chromatin is seen as basophilic material in the nucleus. It may be located adjacent to the nuclear envelope. It consists of DNA and its associated proteins, called histones, in a condensed state. If the DNA is placed end-to-end, this would be nearly a meter long. Fitting the long DNA into a nucleus requires several orders of coiling. At regular intervals along its length, each DNA molecule is coiled 1.5 turns around a cluster of 8 molecules of the basic protein histone. Each histone and its encircling DNA constitute a complex called a nucleosome. There are two types of chromatin. Heterochromatin, which is inactive chromatin, can be seen in basophilic clumps in the light microscope, and electron-dense coarse granules in the electron microscope. Euchromatin is the less coiled portion of the chromosome and can be seen as slightly stained basophilic areas in the light microscope and finely dispersed granular material in the electron microscope. The nucleus is bounded by a nuclear envelope consisting of two parallel membranes separated by a 20 nanometer space called the perinuclear cisterna. At a few sites around the perimeter of the nucleus, the outer membrane is continuous with the membrane of the endoplasmic reticulum. At many sites around the periphery, the outer membrane is continuous with the inner membrane around nuclear pores that serve as avenues of communication between the nucleoplasm and cytoplasm. It is not uniformly distributed and closed by a pore diaphragm. In nuclear envelopes that have been isolated and viewed at high magnifications, the pores are found to be lined by 8 particles or subunits that projects spoke-like structures into the center so that it looks like a wheel with 8 spokes from the top. At the center is a central plug. Note that in this image, one of the pores appear to be open in the center. The nuclear pore acts as the principal barrier to movement of particles greater than 10 nanometers. The nucleus is visible in the light microscope as an eccentric structure in the nucleus. It is the site of ribosomal RNA synthesis. It stains deeply with the basic dye. The number and size of the nucleolus depends on the cell type. This ends the first part of the lecture. Points discussed were the following: introduction to the use of the microscope in histology, such as types of microscope and images seen, and the units of measurement used. Second is the organization of the cell. The two main cellular components of the cell will be the nucleus and the cytoplasm. Third will be the difference between organelles and inclusion bodies. And lastly, I have talked about the histology of the nucleus and its components. Please view the second video on the continuation of the lecture on the cell.